Um, Great. This is going to be fun. I want to welcome everybody and encourage you to come in. Please, there are lots of seats down in the front. It seems like there are a few people that remember Ron DePino and are eager to welcome him back to Dana-Farber. It's a pleasure to welcome Ron. And today, Ron's going to be delivering the Emily Frederick DiMaggio Memorial Lecture. The Emily Frederick DiMaggio Lecture is an annual seminar in basic science that was founded in 1978 through a gift made by Emily and Dom DiMaggio. Emily was a longtime trustee, donor, and fundraiser who devoted her remarkable energy and talents to advancing many Dana-Farber's charitable missions to benefit children. The late Dom DiMaggio was a 7-time All-Star and 10-year veteran of the Boston Red Sox and longtime teammate of Ted Williams. We're forever grateful to the DiMaggio family for their generosity and support, and to Emily for her commitment and exceptional service to advance Dana-Farber's missions over the years. We are deeply honored to hold this seminar, which bears her name. So we're pleased to have Ron here. I read some of the fine print for this lecture, and it turns out there is one small thing that Ron is going to have to do in order to be able to be awarded the plaque here, which is that Ron was born in the Bronx and is a lifelong Yankees fan, but he's promised me that he's going to give up all affinity and affiliation with the Bronx baseball team, not to be named, and he'll be pleased to accept the DiMaggio Award. Okay. All right, so I'll be quick. I'll give you a little background on Ron. He's from the Bronx, educated at Fordham College, then Albert Einstein College of Medicine, where he got his MD with distinction in 1981. He did his internship and residency at Columbia, and then was a postdoc at Columbia with Fred Alt, who's here in the audience. He went back to Einstein, rose to the rank of professor, and then in 1998, we were fortunate enough to bring Ron up to Dana-Farber, and he was with us until 2011, when, of course, he left and was offered the presidency of MD Anderson. He's currently the Harry Graves Burkhardt III Distinguished University Chair. I'm not going to go through all his honors. He has all the expected academic honors that you would imagine. I will just mention all of the academies and things like that, but I'll mention just some of his more unusual honors. He has a knighthood from the Republic of Portugal. Actually, he corrected me and told me that he's now a commander, which is more important than a knight, for the Order of St. James of the Sword. And he is also the science advisor to the Pope, which I'm looking forward to hearing more about tonight at dinner. He's, of course, famous for his work on telomerase. He's touched on many interesting areas in metabolism, sex differences in cancer and aging. But today, he's going to come back home and talk about pancreatic cancer, where he did amazing work here at Dana-Farber and trained some remarkable people who are still in the Harvard system. Please welcome Ron.
Well, it is indeed a pleasure to be back home. I have very fond memories, friends, lifelong friends, and of course, one of my life mentors in science, not just Fred, but David Livingston, and it's just with a heavy heart that he's not here to ask questions that I can't possibly answer. He was really, truly impactful in my career, and the privilege that I had of coming to this institution was really because of David And a lot of the successes and so on, the opportunities that I've had were really driven by his generosity and his mentorship. So I truly, truly miss him, as we all do. Well, today I'll be talking about pancreas cancer, and I wanted to do that because I wanted to celebrate some of the work of folks in the audience here today. And I think we've reached a point in this disease where we can make a decisive assault on this problem. Before I get into that, I wanted to just give you a little bit of background just to frame how we think about cancer and what the lab is particularly interested in. On the left side here, we've been very interested in trying to understand driver oncogenes. What are the genes, the genetic elements that drive the development of cancer and how they affect cancer hallmarks? It's more from a cancer cell intrinsic point of view. But we've also studied the anatomy of the genome and identified novel strategies to be able to expand the cancer target pool, concepts such as collateral lethality, which Bill Hahn's lab also identified and has studied. So I'll talk very briefly about one example of that. But I'll spend most of my time on the right side, which is to really understand how those genetic elements in the cancer cell influence the biology of the tumor. And what we've now learned is that those genetic elements in the cancer cell can very deliberately and specifically influence the composition of the tumor microenvironment, the kind of heterotypic interactions that occur between cancer cells and cells of the tumor microenvironment, and in turn, how those cells provide trophic support, not just merely immune sequestration and so on, but really create these symbiotic circuits that themselves are therapeutically exploitable. And so the first indication that actually these genetic elements, these driver oncogenes in cancer are relevant to tumor biology came with a model system that Linda Chin developed. And she developed the first inducible oncogene model of cancer. And in this case, it was driving a TET-inducible RAS allele in a melanoma model system that it was INCARF deficient. And she wanted to ask the question, we knew that RAS could create melanoma, but was it still important against the backdrop of many other genetic alterations that occur in the life history of these cancer cells? Is it still rate limiting? Is it still important? Is it still a tumor maintenance gene? And so when she flipped off the RAS switch in these fully established tumors, she found that indeed it was a tumor maintenance gene, and this led to the concept of driver oncogenes, or as Bernie Weinstein coined it, oncogene addiction, so that this was the experimental evidence for that. But the most interesting thing in this story was that within hours of flipping off the RAS switch, there was endothelial cell apoptosis. And this taught us that RAS in the cancer cell compartment was actually instructing the host to grow new blood vessels. And of course, a lot of this has to do with the seminal work that Bill Kalin has since articulated with hypoxia control. But we also now know that RAS itself regulates factors such as VEGF that can support neoangiogenesis in the tumor cell compartment. So this then set us on a path to try to understand how genetic alterations in cancer cells influence the tumor microenvironment and turned us into a lab of tumor biologists. And so I'll be telling you mostly about our work in pancreas cancer. And this actually, we were inspired to work on this because I attended an NCI think tank in which we wanted to understand what made this disease such a tough beast. And not much was known at the time, so we thought that perhaps some science could illuminate therapeutic strategies to impact this dreaded disease. Not much was known, except there were some signature alterations that occurred, most prominently KRAS, which occurs in the overwhelming majority of pancreas cancers, and the tumor suppressors that you see here, INCARF, P53, SMAD4, as being very common. And a real breakthrough in the field came from Dave Tuvison and Tyler Jax's lab with the lock-stop-blocks KRAS allele that allowed targeted expression of RAS into various tissues, in this case the pancreas, and that led to the first mouse model for pancreas cancer. And what Nabil and Andy Aguirre did was to layer in these tumor suppressor genes, and they developed the first mouse model for advanced pancreas cancer. And this then afforded us with the opportunity to study the role of these tumor suppressors and KRAS in influencing the biology of these cancers and so on. Now, what we were particularly interested in is trying to understand the function of RAS. And so we, again, developed an inducible model system to study that. And this is the work of Alec Kimmelman, who is also here at the Farber, and Hao Chung Ying, who now are leading very important programs in their own right. And what they did was to develop a TET-inducible KRAS allele, and did in a fully established tumor. You could see that in these tumors, they're very PET positive, they're very glucose avid, they produce an enormous amount of lactate, a couple of orders of magnitude more lactate in the tumor microenvironment of pancreas cancer than you see in an exhausted muscle, which is profoundly immune suppressive, and I'll get back to that towards the end of the talk. But essentially what they found is that when they flipped off the RAS switch and did multi-omic analysis, so transcriptomics, proteomics, metabolomics, what they found was that the main function that RAS had in a fully established tumor was to maintain anabolic metabolism. And you could see here that within switching off the KRAS switch, these tumors now become PET negative. And so then we went on to ask a number of additional questions, which is, well, how is it that it regulates glycolysis? And we found that there was transcriptional upregulation of genes that are in the core axis for glycolysis, as well as genes that are involved, for example, in the non-oxidative arm of the pentose phosphate shunt that generates nucleic acid building blocks, also enhances glycosylation and so on. And so this basically established that KRAS really generates a highly glycolytic state to really fuel anabolic growth of these cancer cells. Now, one of the things that we were interested in is seeing that these genes were transcriptionally upregulated. We wanted to connect oncogenic KRAS to the regulation of these genes. We found that MYC binding elements were enriched in these gene targets, but were unable to forge a direct biochemical link between KRAS and MYC in pancreas cancer cells. And so we were really uncertain as to how this played out until we did a series of experiments in which we examined growth factor signaling circuits in the context of KRAS signaling. And one of the interesting things that we found was that when we flipped the RAS on and off, we regulated the expression of two cytokine receptors, IL-4 and IL-13. And what was particularly interesting about that is that the ligands for that were not coming from the cancer cell, but rather from invading CD4 cells that constitute up to 50% of the cells in early stage pancreas cancers. And those cells, those Th2 positive cells, were secreting IL-13 and IL-4, which then activated JAK1 and STAT6 to sit on the long upstream enhancer element of MYC and upregulated MYC to turn on the glycolysis genes. And I wanted to just present this example because it's yet another example that even something as hardwired as oncogene-driven metabolic reprogramming, you would think is cancer cell intrinsic, but in this case, it requires this heterotypic circuit between CD4 cells that are providing ligand to close the loop on a circuit to regulate now these glycolysis genes. And so if you wipe out those CD4 cells, you dramatically decrease glycolysis gene expression, hexokinase, so on and so forth. If you also interdict this pathway at multiple levels, at the level of the receptor, or JAK, or STAT6 at the genetic level with RNAi, you can also dramatically decrease the anabolic metabolism, and you dramatically decrease the tumor size and increase the survival of the mice. So again, just using this as an example to establish the importance of the tumor ecosystem for this critical hallmark of metabolic reprogramming in pancreas cancer. So next, with this oncogene-inducible model, it gave us the chance to also ask the question, is KRAS a tumor maintenance gene? And what you can see here is that when you flip off the RAS switch, indeed, a lot of the animals, about a third of the animals actually are cured. They survive. But 2/3 of the animals still succumb. A third of those, half of those are basically DOX-independent dysregulation of the KRAS transgene, so that's boring, so they re-express the KRAS transgene. But the others are true ERK-negative KRAS-independent escapers. And we also did transcriptomic genomic analyses, and what we found, which was similar to what Bill Hahn showed in lung cancer, is that when you have these KRAS-independent escapers, some of them have YAP amplification, which is also YAP dysregulation is seen now in human cancers, patients that have been treated with KRAS inhibitors. You also see RTK activation, and this also nicely parallels the beautiful work that Andy Aguirre's lab has done in examining the escape mechanisms in human pancreas cancer under KRAS therapeutic perturbation. And so this has led to efforts to try to use KRAS inhibitors in pancreas cancer. Now there are 17 pharmaceutical companies that are developing TEAD inhibitors as well. And just in the interest of full disclosure, one of the companies I've established is developing a TEAD inhibitor. And so this was interesting because it suggested that as important as KRAS is, there are other bypass mechanisms that allow for KRAS-independent growth. And so we're very interested in those mechanisms, and we've cataloged at least YAP and RTK dysregulation. But there were a bunch of others that did not show any genomic changes. And so we were very interested in doing that. And we noticed that in a lot of these cancers, there seemed to be a change in cell state. And so we suspected that maybe epigenetic dysregulation might be operative in those as well. So what we then did, and that we in this case is Ping Hao, is to take this KRAS-inducible cancer cell system and introduce a couple of hundred orpheomes for epigenetic regulators, and then put them back into the mouse in the absence of doxycycline to look for KRAS escapers, KRAS-independent growth. And number one hit in this was HDAC5, and this was a pretty robust escape mechanism, that is HDAC5-driven KRAS-independent growth. But what was most interesting about this hit is this only worked in vivo. It did not work in vitro, which immediately pointed to potentially the host as being a co-conspirator, collaborator in HDAC5-driven cancers. And so we looked for ligands and looked at KRAS versus HDAC5 escapers. There were 13 ligands that were different, that were growth factor ligands. And one ligand, TGF-beta, was capable of rescuing in vitro proliferation of these HDAC5 escapers. So then we examined what the potential source of TGF-beta might be in the context of these tumors. And one of the most striking things that we found in these KRAS escapers is that there was a dramatic shift in the tumor immune composition in these tumors. So in the KRAS, it's dominated by granulocytic MDSCs, whereas the HDAC5 escapers, that population is largely gone, and it shifts to these tumor-associated macrophages. And these tumor-associated macrophages are indeed the major source for TGF-beta in these HDAC5 escape tumors. And if you use clodronate to wipe out those macrophages, you're no longer able to support these HDAC5 escapers. And so we've done a lot of work to understand how this circuitry comes about. This is published, so I'm just going to summarize what we've learned. So first, when you suppress RAS or even inhibit the MEK pathway, you get de-repression of HDAC5. HDAC5 is then part of a repressor complex that represses the expression of the suppressor of cytokine signaling. So now what happens is you de-repress the expression of specific cytokines, and the two most prominent ones are CCL2 and CCL7, which are major recruiters of tumor-associated macrophages through the CCR2 alpha receptor. This then recruits these TAMs into the tumor microenvironment. They dump TGF-beta, which signal through the receptor and through SMAD4. And if you knock out SMAD4, use neutralizing antibody against CCL2, HDAC5 inhibitor, CCR2 alpha inhibitor, or TGF-beta receptor inhibitor, you can abrogate the ability of these HDAC5s to grow efficiently in these mouse models. So I use this again as yet another example. In this case, it's the escape from a therapeutic intervention, that here the escape mechanism wasn't a cancer cell intrinsic mechanism like YAP or RTK dysregulation in the cancer cell, but actually is using, in a collaborative way, the tumor microenvironment to provide trophic support, in this case TGF-beta, for KRAS-independent growth. Okay, so now another very important component of the tumor microenvironment are the carcinoma-associated fibroblasts. And this is a very prominent feature of pancreas cancer, which about 90% of the tumor mass is constituted by these carcinoma-associated fibroblasts. So then we ask the obvious question. We, in this case, is a gifted graduate student, Jianchun Hong. And what he did was to do a series of experiments that were just published. And in these experiments, he took conditioned media from carcinoma-associated fibroblasts, and then in the KRAS off setting, he asked the question, what would happen if without conditioned media, and you could see that you get less colony formation, and with the conditioned media from these primary calves or from an immortalized calf line, he was able to support some measure of KRAS-independent growth. And then he also did an in vivo experiment where he co-injected said calves together with the inducible KRAS model in the absence of doxycycline. And I think you could readily see here that with the co-injection of calves, these two lower panels, you're able to support tumor growth, but in the absence, you get very meager tumor growth of these lines. And this is all in the absence of doxycycline, so KRAS is off. So these calves are able to support some degree of KRAS-independent tumor development. So then the question is, how is this happening? And here again, he did multi-omic analyses and found very interestingly that the PI3 kinase pathway, Luke Hanley's favorite pathway, and Tom Roberts is activated. And then he did RTK arrays and found that ERB2/3 were dramatically upregulated. They were activated and transcriptionally upregulated about 5 to 10-fold upon extinction of KRAS. That's in the cancer cell compartment. And the source for the ligand for these RTKs, this heterodimer, which is NRG1, is found in the carcinoma-associated fibroblasts as well as in myeloid cells. But all the different calves, iCalves, myCalves, antigen-presenting calves, they all express NRG at some level. And so then the obvious thing was to see if we could interdict this pathway either at the level of NRG or at the level of inhibition of the receptors to see if we can improve the responses to KRAS inhibition. And he went on to basically show with RNA in situ hybridization that the PDPN positive calves are all expressing the NRGs. The antibodies just didn't work on IHC, so we had to do RISH. Okay, so now he wanted to do a series of genetic experiments. So he used calf conditioned media or recombinant NRG in the absence of KRAS signaling to show that both of those additions is able to increase colony formation. And then what he did, he CRISPR knocked out ERB2, ERB3, or ERB2/3, and was able to abrogate colony growth. So the receptors are important. And then we got an antibody from Genentech, the anti-NRG antibody that was developed. It failed in clinical trials due to neurological side effects, so it was never developed. But this antibody works quite well short-term in these rodent models. And I think you can see here that when you add the NRG antibody, you're able to quell the impact of tumor growth with the co-injection of KPC and CAF in the absence of doxycycline. And so this was, in the presence of doxycycline. And so this absence of doxycycline. And so clearly NRG was an important component of this CAF ability to maintain tumor growth in the absence of KRAS signaling. And then he finally did a series of experiments that with the intent of providing a clinical path. The first thing he did was to genetically introduce a hairpin for ERB3. The red line is just KRAS inhibitor. This is the MRTX inhibitor, G12D inhibitor. You can see an increase in survival. And with the depletion of ERB3 plus the inhibitor, you see a further increase in survival. In the absence of KRAS extinction, the depletion of ERB3 is irrelevant because it's actually expressed at low levels. It only gets induced upon extinction of KRAS. And then here, what he did was to use the MRTX inhibitor with the NRG1 antibody, and again was able to increase the survival of these animals above just KRAS inhibitor alone. And then finally, in a highly clinically actionable combination, he used pertuzumab, which targets the receptor complex, together with MRTX113, and it was able to increase the survival of these animals. And so this provides a good clinical path hypothesis for KRAS inhibitors, where the responses to KRAS inhibitors have been a major breakthrough for pancreas cancer, but unfortunately, like most targeted therapies, there is rapid recurrence, and this at least will provide more runway for our patients. So essentially, what we see is that when we extinguish RAS either genetically or pharmacologically, we get this 5 to 10-fold increase in the expression of ERB2/3, and then CAFs, as well as some myeloid cells, are able to provide the ligand NRG1 to then activate this receptor and presumably activate the PI3 kinase AKT pathway. We saw this being activated, but we haven't formally proven that that downstream signaling surrogate is really what confers KRAS independent survival. So that's something we still need to do. So again, just providing this as yet another example of how these collaborative interactions, symbiotic interactions within the tumor microenvironment can provide escape mechanisms from KRAS's very important role in sustaining pancreas cancers. So now what I'd like to do is to just spend 1 or 2 slides on just another concept and then spend the rest of my time on the combination of immunotherapeutic interventions and KRAS inhibitors. But I did want to mention this, and this is something that Bill Hahn has also championed, this concept of targeting tumor suppressors. And the idea was that when you look at the anatomy of a deletion event in a tumor suppressor locus, it is not just a surgical deletion of the tumor suppressor gene, but you're also deleting neighboring genes. And a lot of these genes are basic housekeeping genes. But cancer cells can survive because there are paralogs elsewhere in the genome, providing a level of functional redundancy to enable those cancer cells to still survive. But it provides a cancer-specific vulnerability by targeting that paralog. And so we have a number of examples of this, but the one example that I wanted to give is of the SMAD4 locus, which in that locus we have mitochondrial malic enzyme 2, and there are 2 mitochondrial malic enzymes, 2 and 3, Mitochondrial malic enzyme 1 is in the cytoplasm, and it's cell essential. And essentially, it's a very commonly deleted gene in cancer. You can see here, these are SMAD4 deletions and ME2 deletions. And in pancreas cancer, about a quarter of pancreas cancers are homozygous null, homozygous deletion for SMAD4. And Me2 is also co-deleted in these. And there's about 100,000 newly diagnosed cancers that are SMAD4 homozygous deleted and also MAD2 deficient. And so what P.J. Day did was to design an inducible hairpin system to knock out the remaining mitochondrial malic enzyme. He got some very nice hairpins that was able to deplete, and he also did rescue experiments, Bill, okay? And showed that these were specific. And so with this depletion, he was able to show that this completely abrogated the ability of these cells to form tumor growth. So the depletion of malic enzyme 2 and 3 really dramatically abrogated the ability of these cells to maintain their tumorigenic potential. And this is published, so I'm just going to summarize what he learned. What actually got this paper published in a very nice journal wasn't just the collateral lethal relationship, but the discovery of new functions for malic enzyme. It was known that it was very important for generating pyruvate and feeding the TCA cycle classically, but also what was found was that it played a very important role in maintaining reducing potential in the mitochondria, so it generates an enormous amount of NADPH. And in the absence of mitochondrial malic enzyme, there is a dramatic increase in ROS and mitochondrial. The mitochondria get fried, and the cells undergo apoptosis in a Cas3-dependent manner. But the other interesting thing that he found was that this high ROS state activated AMPK, which is important in repressing the expression of this transcription factor SRBP1, which is a major regulator of BCAT2. And this is very important for converting alpha-ketoglutarate to the building blocks for de novo nucleotide biosynthesis. And in fact, this is so important that if you overexpress BCAT2 in the cell line that I showed you earlier, that where you don't form tumors, you can rescue the tumorigenic potential of malic enzyme 2,3 null pancreas cancers. And so this is a new mechanism for mitochondrial malic enzyme to fuel de novo nucleotide biosynthesis. Okay, so I just wanted to use that as an example. There are now quite a few examples of collateral lethal targets. There's actually a couple of pharmaceutical companies that are developing malic enzyme 3 specific inhibitors. I think this basically expands the target pool of cancer genes that we might be able to go after. And the nice thing is that it provides a very strong clinical hypothesis, a patient responder hypothesis, because obviously you would select the patients that have the deletion of the paralog to enlist into the clinical trial. Okay, so now for the main thing that I want to talk about, and this is mostly unpublished work, is really trying to understand the role of the immune system in supporting tumor development in pancreas cancer. And so this has been a real disappointment in the field. It's been completely unresponsive to standard checkpoint blockade, such as CTLA-4 and PD-L1, et cetera. Even the combinations have yielded very meager responses. Only one of 35 patients responded to dual checkpoint blockade. And so Pat Gulhati, a physician scientist lab, suspected or hypothesized that it could be perhaps an alternative T-cell checkpoint and or the role of myeloid cells in providing immune suppression in the tumor microenvironment, where these cells are very prominent in the tumor microenvironment. So what he did first was to do single-cell sequencing in collaboration with Nick Navin at the time, and cataloged all many different immune regulators. And you can see here in that the exhausted CD8s, there are a couple that came up. One was PD-1, another one was LAG-3, 4-1BB, and TIM-3 were expressed in the exhausted CD8s. So, and this was also confirmed in human PDAC specimens, both naive and treated patients, where a very high percentage of them expressed LAG-3 and 4-1BB by immunohistochemistry, about 90–80%. And from the single-cell data, published work from Nina Steele and others, we see that these molecules are expressed in the CD8 positive compartment. So then what Pat did was to do a series of clinical, preclinical, experimental therapeutic trials in which he took three independent orthotopic lines, syngeneic lines, put them into the pancreas, generated sizable tumors, and then treated those mice with a single shot of gemcitabine, which is standard of care to create some degree of immunologic cell death, and then did a 4-week treatment course with a whole series of antibodies, some of which are listed here. And you could see that PD-1 and CTLA-4 had no response, similar to what you see in the human situation. But 4-1BB and LAG-3 had signal of activity. Note that TIM-3, which was expressed, did not have any activity. So this then led to a survival study. And again, this is just transient 4-week treatment. And what he's found was that this tumor response was associated with a prolongation in survival. So this was eye-opening because in the 20 years that we've been using this model, nothing has worked. It's just been very, very low responses to many different therapeutic agents. And if you combine 4-1BB and LAG-3, which have different mechanisms of action, you get additive survival, but the animals still die. So the question is, what else can we target aside from those T-cell checkpoints? And so here we looked at, whenever we did these studies, we would perturb the system and then audit the composition of the tumor microenvironment. What are the adaptive mechanisms and so on? And one of the things that we found was a strong prominence of a neutrophilic granulocytic MDSC signature in both mouse as well as in human. We developed about 10 years ago a 67-gene signature that enabled us to identify an approximation or equivalence of neutrophils in the human samples. And so we use this to catalog the different cell compartments and so on. And we see that this is prominently represented both in mouse and in humans. So then we looked at TCGA data with our signature. And you can do the same thing with other immunocytes. And indeed, with CD8 T cells, you get a statistically significant, but I would say marginally biologically significant difference in outcomes. But this was telling us that these MDSCs in humans have a very prominent role to play in the biology of these cancers, with a high MDSC signature having a very poor survival and a much better survival in the context of a low MDSC signature. And again, from single-cell data from Nina Steele and others, we can see that these MDSCs are prominently represented, and these MDSCs express CXCR1 and 2 cytokine receptors. And so then what Pat did was he used a series of agents, and I'm showing you here the one that was most prominent and effective. He used the Syntrex CX6A2 compound, which is a CXCR1/2 inhibitor, along with LAG-3 and 4-1BB. And in 3 independent PDAC orthotopic models, he was able to eradicate disease in 90% of the animals. And this is just the dual checkpoint blockade showing a very significant increase in survival, but ultimately the animals succumb. So the triple therapy of LAG-3, antagonist antibody, FOLN-BB agonist antibody, and the CXCR1/2 inhibitors gave unprecedented survival benefit in the orthotopic setting. And then he went on to show that these animals had immunologic memory. He re-challenged these animals with the same cancer cells, and those in the absence of therapy, all of those animals were able to eradicate those cancer cells, whereas the controls all succumbed to the disease. Okay, and then the other thing, and this is a point that I really wanted to emphasize, we do work with orthotopic tumors, the field does, but we also examine things in orthochronous tumors, naturally arising tumors. You can imagine with an orthotopic transplant, you've got injury at the time of injection. Those cells are never syngeneic. There could be enough immunologic neoantigen differences that have accumulated, et cetera, in these cell lines. And so we always like to confirm things with the orthochronous setting. And this is a very high bar because when you turn on RAS in these tumors, the entire pancreas becomes a cancer. And it's multifocal, multiclonal. So it's a very, very tough disease to control. And so in this setting with LAG-3, FOLN-B, and CXCR2 inhibitor, we're able to increase the survival of these animals. About 20% of 2 of the 10 animals had no evidence of disease at the time that we had to publish the paper and to sacrifice the animals. So there was no evidence of disease, but we don't know if there's minimal residual disease, and it would have come back, and so on. But nevertheless, this is unprecedented in the context of orthochronous tumors, but it's a big difference between orthotopic injections and these naturally arising tumors. So I thought I would emphasize that came up in earlier discussions today. So now, understanding the role of KRAS in these pancreas cancers also gave us ideas to potentially expand this combination by targeting both KRAS and these various points in the immunity cycle. And this was particularly important because, as I mentioned earlier, KRAS makes these cancer cells very glucose avid. Remember that the PET that you see here. And they also generate enormous amounts of lactate. This is profoundly suppressive for anything that wants to kill a cancer cell, a CD8, dendritic cells. They really thrive on glucose. Work from Jeff Rathmel and Jeff Rabinowitz. And also, lactate is fuel for Tregs, NDSCs, M2-like macrophages, and so on. So it stands to reason that inhibition of RAS might create a more hospitable environment for KRAS inhibition, for the tumor immune microenvironment. So on the left side here is similar to the experiment that I just described, which was single shot of gemcitabine, multi-armed clinical trials, MRTX and triple, or MRTX and PD-1, which is the backbone for almost all immunotherapies in pancreas cancer and other cancers. And so what's particularly exciting about this data is that 1/3 of the animals do not have evidence of disease 6 months out. And we're still studying the adaptive responses that have occurred in the ones that did succumb and did recur. But first of all of them had complete responses. And by the way, these are very, very large tumors, the equivalent of a football in a human. In fact, the controls you can see here, all of the animals are dead within 1 to 3 weeks. And even the triple immunotherapy, which is the next line over, didn't have a chance to catch up and enlist the immune system. And so you get a slight increase in survival, but the animals largely succumbed. With KRAS inhibitor, those are these middle lines here, the red line. You see an increase in survival, and many have reported, like Andy and others. And here, the addition of PD-1 has no impact. I think that's important. It's a recurrent theme that we've seen in orthochronous tumors, whereas it responds with orthotopic injection tumors, but not in the orthochronous setting. And this is just an example. On the right side is where we genetically extinguished RAS. And we did this because we wanted to see, well, what if we had an even better inhibitor of RAS? Could we have even greater disease control? So here we're genetically eliminating, extinguishing the expression of oncogenic KRAS, and you can see that instead of 36%, we now see 67% with the triple immunotherapy. This is the data that I showed you previously, where a third of the animals have no evidence of disease and just by extinguishing RAS. But the combination of the two really elevates the tail and provides very long-term, this is 300 days out, very long-term survival of these animals. So I think there's room for improvement in some of the KRAS inhibitors, and there's a pipeline that's entering into clinical trials that we all hope will do even better than some of the initial drugs that have hit the clinic. And this is just MRI study. This is just showing you, you have these huge tumors. They all undergo complete regression. And then these this is KRAS inhibitor and triple IO. This is an example of a recurrence. And this is an example where, again, you get a response and no evidence of disease half a year out. And these are all, KRAS and PD-1 plus KRAS inhibitors. They all respond initially, and then they all recur. And so you can see 4 of the 11 animals have no evidence of disease at this point by MRI. So now looking at these tumors, we look both in the tumor core and the periphery. And so you get lots of immunologic responses that are occurring in the periphery of these tumors when you do these immune modulatory experiments. The key is what's going on in the core of the tumor. And so here you could see that it's a desert, and my computer just went out. So it's a desert. You don't see much in the way of T cells. You have, with the KRAS inhibitor, more T cells coming in, and a couple of those T cells are now proliferative. With the triple IO, you get T cells, but a lot more proliferation. And with the triple IO and KRAS, you now have the ability of these T cells to proliferate. Again, you have more glucose availability, less lactate, and so you have these Ki-67. And all these other immunocytes are reprogrammed in a favorable way. And of course, we've done a lot of different analyses, and I'm just going to show you one slide, which is looking at the CD8s in this particular case by single cell. We could see that all of the different CD8 subpopulations are there, naive, effector, and exhausted, as well as CD8 proliferating cells. And you could see across panel D here all the different therapies that we used: control, triple, MRTX, MRTX plus triple, or MHS plus PD-1. And I think you could easily see how the KRAS inhibitor and triple expands all of the CD8 populations, and also, most importantly, the proliferating CD8s. And they have the usual killer markers on them as well that are at the top panel. And this is just quantitation of the numbers of different CD8s in the context of the triple. It's interesting that you actually see less proliferating T cells when you combine with PD-1 and KRAS inhibitor relative to KRAS inhibitor alone. And I just wanted to show you this one immunohistochemical slide. This is what the KRAS inhibitor and triple looks like. Virtually every CD8 is associated with a cancer cell. And in the context, and this is after 2 weeks of treatment. And here you could see that PD-1 and MRTX has some T cells, but it's fairly meager in its immunological response. And again, this is the backbone that many are using, and I emphasize that I think it'd be worth exploring alternative checkpoints in this case, like LAG-3. So here's the summary. So I told you that KRAS is super important in pancreas cancer, and its primary function in pancreas cancer is to regulate anabolic metabolism. Now, we've done this in other cancer types, For example, in colon cancer, KRAS does not regulate metabolism. Instead, it regulates immunity. So same oncogene, very different biology in those two GI cancers as just one example. And then I showed you that there are multiple cancer cell intrinsic, like YAP, RTK mechanisms that can confer KRAS bypass in the setting of KRAS inhibition, but also non-cell autonomous mechanisms that involve host, such as macrophages and CAFs, that provide trophic support, and all of these are clinically actionable. I also show you that KRAS inhibition synergizes with this triple immunotherapy that was rationally developed based on therapeutic perturbation and auditing regulatory circuits. It's well tolerated. Obviously, it's going to be a high bar for human studies. We see the transient elevation in LFTs, as you see with some of these agents in humans, but this is going to have to be sequenced, I would imagine, and that these targets are all expressed in human PDAC specimens, both naive and treated. And I briefly mentioned collateral lethality as another strategy to expand the cancer target pool in pancreas cancer and in other cancers as well. These are the folks that did the work. I mentioned their names along the way. It was all of these folks trained here at the Farber, and they have, now are running their labs and are huge success stories despite the lack of mentorship. And including, I didn't mention actually, notice Ned's name up here. Ned obviously had a very celebrated career and served the community with great distinction. Did some of the early P16 and ARF crosses to show that they were important, both important in pancreas cancer as well. And then some of the recent work are the folks that are highlighted that I mentioned along the way, and the folks that have supported us, and mostly this community, which has taught me an enormous amount about cancer and how to do science. So thank you for your friendship and mentorship over the years.